it's not possible to follow individual fibers all the way from their origin to their destination. And it's very low throughput. It's very time consuming and labor intensive to do this even for a single brain. And you cannot do this to get a full connectome of the brain, because as you dissect one pathway, you have to destruct all the ones that are crossing with it. However, for a long time, this was the only way that was feasible in the human brain to get information about what is connected to what, and how we have some rudimentary knowledge about what are the main pathways, fiber pathways in the brain. And the other source of information comes from tracer injections, which are not feasible in human subjects. So these are done in non-human primates, and preferably in non-human primates that have good homologies with the human brain, like macaques, so old world monkeys. This has very high precision. So a tracer is injected in the gray matter, and then it labels all the fibers that are connected to that specific location in the gray matter. It is also low throughput, so these are also very difficult studies to do. And for a long time, it was only possible to do this for, let's say, a handful of different injection sites in a same brain. So again, it won't give us a whole brain connectome. However, this was for a long time the most precise way, and it still is actually to this day, the most precise way to determine what is connected to what in the non-human primate brain, and then sort of extrapolate based on anatomical knowledge to the human brain. So actually, most of what a surgeon, the knowledge that a surgeon has when you're in the OR and they're about to stick a sharp object into your brain, and they have to figure out what they should and shouldn't cut, comes from these kinds of studies to this day, not from diffusion MRI, actually. So obviously, when diffusion MRI came around, based on these fundamental ideas and observations of the mid to late 90s, it became the first way that we had to actually do this kind of dissection non-invasively by sensitizing the MRI signal to the diffusion of water molecules along fiber tracts, and then following those principal directions of diffusion and connecting the dots across the brain. So one of the first questions, of course, was, okay, how does this compare to those classical anatomic techniques, and how accurate is it? And so from those very early studies that compare diffusion tractography to the two classical sources of information that I showed you earlier, so gross dissection in non-human brains here at the bottom, and tracer studies in non-human primates up at the top, it became clear that diffusion tractography can give us, overall, has a good chance of giving us these main highways of the brain. But in order to get those highways correctly, there was a lot of manual interaction to clean out the things that were not correct. And so actually the first way that histological ground truth was ever used in tractography was these very simple early virtual dissection protocols, where based on this anatomical prior information, the authors came up with ways to define brain pathways in diffusion tractography data based on a small number of regions of interest that a certain pathway should or should not go through. And so that was the first way to dissect these fiber pathways virtually using diffusion MRI data. And of course, at first, this was manual. So these ROIs had to be drawn manually by someone who had a good knowledge of the anatomy. And then there was a big push to automate this process. And related to today's workshop, a few years ago, we came up with a way to do this, to automate the dissection of fiber pathways in a way that leverages high quality data from, at that time, from the Connectome 1 scanner to train an algorithm that then can automatically reconstruct the same pathways accurately in diffusion MRI data of much lower quality that can be acquired in conventional clinical scanners. And this was work by Chiara, who's over here, and she will talk to you in the afternoon about an amazing new tracked atlas for neuromodulation that she's building from Connectome 2 data. But back to this earlier work. This leveraged some prior work that we had done with tracks constrained by underlying anatomy, or tracula, which is an automated approach for probabilistic diffusion tractography, where the shape of a certain pathway is fit to two complementary sources of information, both the subject's own diffusion data and some prior knowledge on what parts of the anatomy, what anatomical structures this pathway should or should not go through or next to in the left, right, anterior, posterior, et cetera, direction. And so it turns out that this approach, because it only learns from the training data, what are the likely neighbors, anatomical neighbors of a certain pathway, is robust to differences between the training and the study data. And so that's why you can train it on data that came from the Connectome scanner and then apply it to data that were collected with a conventional scanner. And so in Chiara's work, she showed that indeed it was possible to reach with this automated approach up to 90% sensitivity, which means that we can get up to 90% of the ground truth manually annotated tract that we get from the high quality Connectome data, but now getting it automatically from much lower quality data. And in particular, with data that were collected with just a single B value and either 64 or 32 directions only, whereas the training data had been collected with 4 B values all the way up to 10,000 and 500 plus directions. And she showed that the accuracy of this approach was much better than the conventional multi-ROI approach that uses just those basic rules with a handful of ROIs. So if you're thinking, why should I collect data on a Connectome scanner if I don't have one at home and I'm not going to scan my subjects on a scanner like that, this is one approach to leverage data from one of these unique scanners to improve the analysis of routine quality data. However, all of that work, including ours and other automated methods for tractography, has focused on these big highways of the brain that have names like the corpus callosum, the uncinate fasciculus, etc. And for a long time, this has been the view of what a pathway is within the human neuroimaging community. However, from anatomists, we know that these pathways are not monolithic structures that go, from A to B, but they are indeed like highways where smaller fiber bundles will merge on the highway, travel along the highway for a part of its trajectory, and then merge off to go to their destination. And so this is an example from a paper from Suzanne Haber's lab, where they dissected using multiple tracer injections in multiple areas, the different fiber bundles that go through one of these large highways. So the uncinate fasciculus that you see down here. So here we see it as kind of a monolithic structure, but in fact, it's made up of many smaller fiber bundles that will merge onto the uncinate fasciculus, follow it for part of its trajectory, and then merge off. And some that will follow it all the way from the orbitofrontal all the way to the temporal lobe. But there's a lot more detail is what we typically think of when we use big of the brain. So that's where anatomy and histology are go to evaluate the MRI to go beyond the highways down to this more complicated picture. And why it's important to have this more complicated picture, because if you find a difference in the microstructure of a pathway somewhere along the pathway, it could actually mean that there's many different connections that could be affected. And so it's important to have this more detailed dissection of the large highways. And so the gold standard, of course, for doing this kind of validation study is and continues to be the tracer injection studies in non-human primates. And so we have been collaborating with Suzanne Haber now for probably something like 15 years, where she sends us macaque brains that have received tracer injections in her lab. We perform high-resolution diffusion MRI over 48 hours typically, and then send back the brain to her, and she does the sectioning and the histology to visualize the tracers. And then we can compare the tractography and the anatomic tracing voxel by voxel to see exactly where tractography goes wrong and where it goes right. An example of this kind of study was an open challenge that we organized a few years ago, and where teams of tractography developers from all over the world were able to compare their different analysis pipelines against the gold standard of anatomic tracing in the same brains. And so they, some of the basic outcomes of that effort was that we came up with recommended best practices for analyzing diffusion MRI data. You can see more in this paper by Chiara from 2022. And also, we identified some common failure modes where areas in the brain where tractography goes wrong consistently, regardless what specific tractography or pre-processing or what have you method you use. And so it turns out that these common failure modes have to do with fiber architectures that are not crossings. So the community has been very focused on resolving crossing fibers, and this is often talked about as sort of solving all the errors of tractography. But in fact, those areas where most of our current pipelines fail consistently are areas where fibers don't just cross, but also branch, fan, turn, etc. So this is a way where linking diffusion MRI with histology can point to new areas for development for us algorithm developers. And then in the human, you can't do tracer injections, unfortunately, but there's a host of new methods, microscopy methods, that we can use. And so in this work by Robert Jones, who was an analyst in our lab a few years ago and now is on to grad school, we used optical imaging in human samples that had received diffusion MRI. And so this is an example of a very complicated area in the white matter that involves a lot of that branching and crossing, et cetera, where we performed diffusion MRI scans that allowed us to generate data with different diffusion acquisition schemes, both grid-based and shell, single shell and multi-shell. And then we downsampled the data from their original 250 micron all the way down to 2 millimeter resolution to see which aspect, is it the diffusion acquisition scheme, the orientation reconstruction method, or the spatial resolution, has the greatest impact in the accuracy of the diffusion orientations when compared to a technique that's called polarization-sensitive optical coherence tomography that gives us measurements of axonal orientations at the microscopic scale. And so interestingly enough, we found that the most important factor was actually the spatial resolution. And the biggest jump in accuracy was when we went from 2 millimeters to 1 millimeter resolution, which is, is a hopeful message because that's something that we may be able to do. And so going back to this idea of, how do we get away from this view of the fiber pathways in the brain as monolithic structures and map them in their full modular detail, this recent work by Chiara that you can read about in this recent preprint shows a study that we did where we cut up multiple blocks from the same brain. And our goal was to reconstruct an accurate map of the superior longitudinal fasciculus, one of those big named highways of the brain. And at first, we thought that when we got to higher resolutions, we would be able to see more of the fibers, to follow fibers all the way from the parietal to the frontal cortex. But actually, we saw the opposite. we saw that when we went to high resolution, these large long-range connections actually start to break up in multiple shorter range connections. And so this diagram that you see here on the bottom left shows you that when you are at a 1.5 millimeter resolution, which is pretty high resolution in vivo diffusion scan, you see more of these long-range connections. Potentially because at that resolution, many of the shorter range connections get merged with each other. Whereas when you go to high resolution that we can reach ex vivo, as you'll see now, we're down to 400 micron ex vivo. Some of these longer range connections disappear. And so this is where the comparison with the histological ground truth becomes critical. And to this end, we were very pleased to see that as we were doing this work, and at that time we could only do it in these small little chunks of brain, the NIH funded the Brain Initiative Connects program. This was a $150 million investment to produce wiring diagrams of the brain at unprecedented resolutions across species, all the way from the nanoscale in the mouse to the microscale in the human. And so this was an opportunity to take those postmortem imaging techniques that can give us the anatomical ground truth and scale them up so that we can do them not just in small chunks of brain, but in large volumes of the brain and even in the whole brain at some point. And so we were pleased to receive one of the 5 comprehensive centers that were funded by the Brain Connects program. And this is the Center for Large-Scale Imaging of Neural Circuits, where we are collecting both mesoscopic diffusion MRI and different kinds of X-ray and optical microscopy in the same brains. You'll see some of the data from the LINK project in the afternoon in the talks that Ting Gong and Kiara Maffei will present. And in the human brain, we start with a diffusion MRI scan in the Connectome 2 scanner at 400 micron resolution, and then the same brains receive different kinds of optical and X-ray microscopy. So the ultimate goal is to have data that will allow us to train machine learning algorithms to perform more accurate inference of fiber architectures directly from diffusion MRI data. And I will end here by thanking my collaborators, thanking the Brain Connects network, and in particular, the 2 Brain Connects projects that I am a part of, the LINK Comprehensive Center and the APEX Data Coordination Center. Thank you very much for your attention.